Hello everyone, my name is Yelena Gantars and I'm a manager within Scientific and Technical Affairs team. Today I would like to cover a topic accelerating cell culture development through practical applications of innovative technologies. First, a little bit about myself. I'm an analytical development SME with over 6 years of pharma industry experience. I've earned my PhD in 2014 from University of Missouri. Then I joined Patent Biologics, analytical development and formulation scientist team, where I learned anything about analytics that happens between phase one and commercialization. Um, as a senior scientist of Thermo Fisher Scientific Analytical Development, I had to be also liaison between the technical team and the client. Currently, as a manager of technical and scientific affairs team, I'm responsible to providing technical support for clients and business development team, as well as lead technical development projects within Thermo Fisher Scientific. As an introduction to our topic today, I would like to cover some of the changes that happened in the 10 years on the drug market. As we know, about 10 years ago, biologics constituted only 25% of all of the drugs on the market. While recently it has been reported that biologics constitute now a much larger part of the drug pipeline, about 42%. Previously, most of the biologics were discovered, developed, and produced by big pharma, while right now most of those molecules are held by the small and emerging pharma. Another big change in the market that happened within the last 10 years is that there's a severe increase in complexity of new molecules. If 10 years ago the biologics market was dominated by IgG1 and IgG4 types of antibodies, nowadays the list of the biologics that are discovered still focuses on simple antibody molecules. But you see more and more bispecifics, Fc fusions, fragments of the antibodies, recombinant proteins on the market. And the challenge with any complex molecules are that you need to invent new tools, you need to develop new cell lines, better upstream and downstream processes, and those are becoming more sophisticated and complex. What does it do to the business? The dynamic definitely changed, and what we do see is that there is a mismatch of drug capabilities in some cases, even in big pharma, with what manufacturing capabilities they have. That means that, or they have to look internally to build new facilities, adjust the old facilities, or train new personnel with appropriate knowledge. Or the final option is to outsource to CDMO. Because there are so many companies in the run to the market, another big change in the business dynamics is that the timeline is becoming shorter and shorter and shorter. And that means that all of our new tools have to be more efficient. They'll have to be better cell lines. And we have to utilize more and more innovative tools and have to keep learning every day. We talked about changes in the pipeline and drug market, but right now I want to touch on the production of biologics changes. While before everyone was chasing the stainless steel bioreactor with its golden standard of 20,000 liter bioreactor, right now you can see that there is increasing number of drugs that is being produced, but the volume that is needed to produce them is decreasing due to all of the innovations in the cell line and production processes. And we are talking about small volumes. We're talking about bioreactors between 2 and 5, so it's blue, yellow, and green on the bottom right bar. In the next several slides, I would like to touch on how Thermo Fisher makes progress throughout the whole developability process to stay on top and ahead of ever-changing demand. The process for pharma services of Thermo Fisher Scientific starts with the development of cell line. As a CDMO, we need to understand what customers are looking for in cell line for their product. Things like high titer, short timelines, a well-documented regulatory-friendly host cell line, as well as low licensing costs are definitely what is driving the business. While these goals in mind, we had worked with our colleagues in bioproduction to develop a new Thermo Fisher host cell line with the following goals in mind. So high titers, we went above 3 gram per liter. We wanted to have a timeline of the from transfection to top clone of between 4 and 5 months. We wanted to ensure that GMP banked cells are in AOFCD medium and that there are no milestones or royalty payments. And the result of this collaboration is Freedom XP2 cell line. Thermo Fisher scientists paired a very robust CHO-S parental cell line with new expression vector system to enhance expression of the proteins of interest. And while these cells are in the final development and will be available on the market soon, we're actively pursuing integration of this cell line into existing pharma services cell line development offering. As mentioned before, pharma services team work starts from the DNA. DNA is introduced into parental cell line through electroporation. After successful transfection, in culture selection is performed in case of CHO-S with pyrimycin and MTX. After that is performed, the cells are introduced into Beacon system. Beacon up-to-fluid platform provides proof of cell line monoclonality and helps us select highest performers by growth and productivity from 7,000 clones that it screens. A little bit more about Beacon we're going to touch on the next slide. After Beacon selection, 24 different clones are selected and moved to the AMBR systems, where the top 4 clones are selected by growth, specific productivity, and some product quality attributes. Then, after narrowing it down from 24 clones to 4 clones, RCB bank is created. Those RCB banks are placed on the stability, 60 generations or 80 generations. After knowing a little bit more about the top 4 clones through the stability and some of the upstream process parameter screening, we have an ability to make a data-informed decision of what is our final clone. As promised, here we're going to talk a little bit more about the Beacon instrument and why Thermo Fisher Scientific decided to include this instrument in the cell line development workflow. First advantage, as can be seen from this slide, it contains 4 microfluidic chips. Each chip contains 1,750 nanopens. And as you can understand, that's ability to screen 7,000 different clones. Another huge advantage of the Beacon is that you can get a titer assessment in the nanopens, meaning that you can choose your cell clones based on the productivity. The last big advantage, and actually the most important one probably, is that the ability of Beacon to provide a proof of monoclonality, which, as you know, you will have to provide to the regulatory agency. It's able to do that because it uses optoelectropositioning technology to be able to move cells within the chip in and out of the nanopen, and its ability to image each nanopen at any time. But let's look how Beacon performs in comparison with other manual workflows. By standard manual workflow, I mean limiting dilution cloning. During limiting dilution cloning, one achieves the duration of monoclonal cell line from polyclonal cell line by a series of the dilutions. To prove monoclonality, one has to image every single well and make sure there is only one cell in the well. And depending on how much labor and cost you want to spend, you can really screen up to about 1,000 clones. After growth here, the 48 top, or maybe however many you want to choose to scale up, clones are selected based on growth only. And then those clones are moved to the scale up to be assessed for the specific productivity of each of the clones. At that point, you will be able to narrow down to top 4 clones. In case of beacon instrument moves cells into each nanopen. Nanopen gets imaged at any time, and this ensures the monoclonality of your cell line. In both workflows, after seeding, cell starts to grow and produce your cell line product. While in manual workflow, as mentioned before, you will not be able to get a tighter assessment. In the beacon, you will be able to get a tighter assessment of your cell line. Therefore, you're getting tighter assessment of all 7,000 clones. This advantage really allows us to narrow down our clones based on following criteria. Titer, which is called AU score for beacon. It's also cell growth, which was assessed by doubling time. Out of those two, you can get a relative specific productivity and assessment of titer on each and a different day, which could be very informative. The other big advantage of Beacon that I didn't mention before is that it automatically excludes nanopens that started from more than one cell. That really shows you the power of Beacon instrument. Therefore, incorporation of Beacon instrument into the cell line development in Thermo Fisher Scientific not only cut down on scientists' manual work and time, providing higher chance of finding a good clone, but also shortened greatly the timeline. Manual workflow takes about 4 to 6 weeks to narrow down to really 48 clones, as you can see on the left side of the screen, while the Beacon workflow, as you can see, can narrow down to 24 top clones within 5 days. And now, after we narrow down to top 4 clones, we're putting it into the AMBR system. And while AMBR 15 system that is showed on the top right corner of the slide is very popular in the industry and in Thermo Fisher Scientific as well to be used for the clone selection and early phase process development, AMBR 15 is not the best presentation of the clone performance in the scaled up bioreactors, be it at bench, pilot scale, or manufacturing scale. Therefore, to overcome that, we decided to incorporate AMBR 250, showed in the bottom right corner, during final stages of the clone selection. Since now the performance of the clones will be representative in the scaled up bioreactors and manufacturing. Even though the clone selection is moving from AMBR 15 to AMBR 250, Thermo Fisher Scientific still actively uses AMBR 15 in the early process development for media and feed screening, as well as parameter screening. Very powerful tool. The AMBR 250 is also used in process characterization work, which if you need a little bit more information, we can provide at a later time. Now cell line development is finished, upstream process is established, and we need to produce the first tax batch and GMP batch. As mentioned in the beginning of the talk, the industry is moving towards the larger number of biologics, smaller scale bioreactors, and because of this factor, single-use bioreactors are becoming more and more popular and becoming a staple of GMP manufacturing. So far on the market, we can find up to 2,000-liter bioreactors that are single use. But market needs also up to 5,000-liter bioreactors, as we've seen from previous slides. This intermediate volume between stainless steel and single-use bioreactor is needed not only because of the volume, but also because the cost of goods is optimal at this stage. Therefore, Thermo Fisher Scientific developed this friendly, scalable from 50 to 5,000-liter bioreactor single use, where 50-liter can be used for tax batches or pilot runs, and 5,000-liter, 3,000-liter, or 500-liter can be used for GMP commercial production. These bioreactors are engineered for robustness, and then one example that I want to present is high cell density cell lines that are so popular today. For that, the additional engineering that was improved on is KLA performance as well as turnaround ratio of the bioreactor. So far, just to summarize, we covered the cell line development and upstream process development, but I'm analytical methods me. What about the analytical support during all of those stages of upstream development? What does Thermo Fisher recommend for analytical? For the product quality attribute monitoring, Pharma Services just introduced multi-attribute method. Multi-attribute method is a low artifact peptide map. The peptide map workflow starts with a Smart Digest trypsin kit which was adjusted for this use. And then the peptides are cleaned up, and afterwards those digested peptides are separated on the Vanquish Horizon UPLC system using reverse phase. Those separated peptides are then analyzed by the mass spectrometer. In this case, we use Q Executive plus hybrid quadrupole orbit trap. This gives rise to the data and a lot of it, which then is analyzed by Chameleon and Biopharma Finder. While Biopharma Finder is used for analysis of new molecules and to ensure that you can identify each and every peak and understand what the peak and what kind of polypeptide it represents in the molecule, Chameleon is there to analyze the data day to day after you've done all of the background work on the biopharma. What does this method provide is really that orthogonal data to glycan analysis, to the charge variant analysis, to the fragmentation analysis that usually was done and previously was done with the conventional methods. And in the later slide, I would like to show you how it compares to every other conventional methods right now. And to go in a little bit greater detail about MEM workflow, as you can see on the right side table, the MEM workflow provides way more information than 7 different other release methods currently used to release drug substance. And what the advantages of MEM are is that it can actually provide information on amino acid level, while most of the other methods used for release are providing you overall information about the molecule. And the difference in the column of MEM between yes and maybe is that MEM is usually run as reduced peptide map, and it will provide you some information about cysteine reduction, disulfide reduction, and trisulfides, while if you're going to run a non-reduced peptide map, you will be able to get more data about that quality attribute. The advantages of the using MEM are lower cost and shorter timelines for our offering. And with that, I want to thank everyone who was able to attend this talk. And our mission is still to provide and enable our customers to make the world healthier, cleaner, and safer. Thank you very much.